previous work which we had last week. I take this opportunity to welcome you again to this lesson. Almost the number is the same as for last week, and even the names the same. Okay. What we are having last week, looked at the experiments of photosynthesis, and we had the whole week and 2 lessons on that. Today, we are yet going to 2 things. One of them will be what is displayed, part of it that is displayed there on the screen. Someone is eating food already. Hurry up, you'll find us on the way. Okay, on eating will get us on the way. Okay, how leaves are adapted to the process of photosynthesis. And so here we want to look at how different leaves have been adapted to ensure they are able to carry out that process of photosynthesis. And the first one we are looking at here, leaves are thin to reduce distance for diffusion. Now, the issue here is leaves being thin. Yeah, when they have a thin lamina, it means that shortens the distance for gases to diffuse. So that one becomes an adaptation for improved photosynthesis. For improved photosynthesis, in so doing, gases can diffuse. If they were thicker, maybe it would shorten, it would make the diffusion surface very bigger, and the rate would not be as fast as when it is thin. So that's the adaptation. Then leaves are broad and flat to provide large surface area for photosynthesis. Yeah, this is to some leaves or plants that would require much photosynthesis. What they do, they have broad leaves which are flattened. And once leaves are broad and flattened, it means that we reduce, we increase surface area over which the process of photosynthesis will take place. So when you see such a such leaves with broad leaves, that means an adaptation to have that process of photosynthesis occur efficiently. Then leaves have more stomata on the lower surface to enable exchange of gases. Yeah, that's what should be. Leaves have more stomata on the surface, on the lower surface, to enable exchange of gases and less transpiration. Can we make that change in the notes? To enable exchange of gases, and then we also reduce on what? Transpiration, because hello? If you are following within the notes, we need to cross-check that statement at the end. The word should be transpiration, not transformation. And then the other one is to enable exchange of gases. So gases would easily be exchanged by passing through the lower point of the leaf. But also it serves 2 purposes. Is that there will be reduced transpiration for that case. Plants would not lose much water in that case. So we have captured the 2 points there. Have you made please note that change? That correction should be made in the notes. Okay, then leaves have waxy cuticle to prevent drying out of the Polysaccharide cells. So we need moisture for this process, and having a cuticle which is waxy would prevent drying out of the Polysaccharide cells. Remember, they are more exposed. If you look at leaves and the Polysaccharide cell, if I can show you this in the picture, you'll be able to see what I'm talking about. Yeah, if you remember, are you able to see that? The internal structure of the leaf?
Yes, sir.
Yeah, so you can see, for example, onto this one, we have this what we call the Polysaccharide or the mesophyll layer. Remember, the mesophyll layer has got Polysaccharide and then sponge. This is 3 and 4, if you can see it clearly. So 3 and 4, the Polysaccharide mesophyll layer has the Polysaccharide cells, okay, exposed here. And so you can see if the cuticle is now, if this is the upper epidermis with the above it, when there is this cuticle, if the cuticle is if the cuticle is waxy, it means it prevents drying out of these Polysaccharide cells which make up the Polysaccharide, mesophyll for that case. Then, yeah, I think this is all what we see from there. Then, if we are to cross-check other things we have talked about, I think shall come back to them when it's time for them. If you need them, shall be sharing and looking at them. Then, if we go back to our next, we say leaves have a pressure to expose them to the sun. You find some leaves have longer petioles than others. So if you find a plant whose leaf special is longer, that's an adaptation to ensure that the leaves are well spaced out to receive what? Are well spaced out to receive sunlight for that case. And it happens with the, if you have seen cassava, you have seen pumpkin leaves, if you have seen quite many plants have petioles, which are even longer, or a petiole, just presence of a petiole. Some plants, of course, have no petiole. Maybe they have the sheath, like those plants which belong to the monocotyledonous group. They don't have petioles. They have a sheath instead. But those which have a petiole is to expose them to the sun. Then the arrangement of the leaves. The leaves are arranged in a mosaic to enable them get exposed to sunlight. I don't know whether you understand what we call mosaic leaf arrangement. Does anyone know what we mean by mosaic leaf arrangement? Yes? Yes, please. Hello? Okay. Someone was asking me the same question that I had already. Someone was asking me the same question that I had already asked. Can someone help us understand mosaic? We say that the leaves are arranged in a mosaic. Now, have you seen the kind of arrangement where I was trying to search for this mosaic? Let me see if I can find how I can demonstrate mosaic leaf arrangement. Hello?
Yeah, teacher.
Yeah, you can follow that. Okay. Have you been able to follow a little?
Somehow.
Okay. What have you understood, Rose?
That it's the arrangement in the same manner.
Okay, now you see, it's all about the pressure of the leaf we have been talking about, eh? That leaves have got different sizes of what? Pressure, okay? So, have you ever seen the cypress plant, the Christmas tree?
Yes.
I do see that on top, they are smaller, but down, the leaves are come a little buried out, okay? You get that arrangement,
Yes.
eh? So it means that the leaves on such a mosaic, the leaves in a mosaic arrangement, the leaves on the lower part of the plant have longer petiole. Then the ones on top have short what? Petiole. So it means that the leaves tend to arrange themselves in a situation where all of them are able to receive the sunlight. Okay? So if wherever you are right now, look at any plant, you'll find that on the topmost plants, okay, they have shorter petiole. Then the ones down have longer petiole. So the arrangement is like the way you see an ant hill, huh? Okay, that kind of arrangement. On top it is smaller, down it is bigger. Okay, so all plants tend to bring in that shape. Okay, if you're drawing a tree, that is what even you think about. Smaller on top, bigger as you get down, which means that there are actually more leaves on the lower than upper, so that when the sun comes, it hits to all leaves. If it was the reverse, if the bigger part was on top, it means that the lower parts of the leaves would not receive the sun. So by mosaic arrangement, we get no, mosaic is not a disease. We're not talking about mosaic as a virus disease, a viral disease of cassava. That is called leaf. That is leaf mosaic. Mosaic is a virus disease which affects cassava. Yeah, it's not the mosaic disease, but this time is the mosaic leaf arrangement in a situation where we see lower leaves having longer petals, upper leaves have shorter petals, so that the arrangement enables all the leaves of the plant to receive what? To receive all the leaves of the plant to receive sunlight for that case. Is it okay now?
Yes, sir.
Okay, somehow.
Yes.
Yeah, somehow it should be okay for us in that case. If I get anything else to describe, maybe I'll share it with you in the meantime. Let's continue to our lesson. Then leaves have branched veins to provide water to all palisade cells. Then the upper side has chlorophyll than the lower side to trap much sunlight. If you have seen in most leaves, the upper side has more chlorophyll than the lower side. Actually, you find that even the upper side of any leaf is more green compared to the lower side or the lower epidermis leaves. I mean, the parts of the leaf on the lower part has less chlorophyll. That's why it's even less green in appearance. Yeah, and then that is all we can say about adaptations of plants to the process of photosynthesis. Do we have any question to that effect? Any question that has can arise from the adaptations you have been talking about? No question. Okay. If there is no question, then we can continue. You can continue if there is no question. Then now we are going to get the next part. The next part of our lesson today, and that is we want to talk about carbohydrates. Carbohydrates. May I ask someone to take us through this work? Let me ask Rosemary. Your mic is on. Rosemary?
Yes, master.
Can you take us through that part of the screen?
Carbohydrates. These are organic food compounds that contain carbon, hydrogen, and oxygen. They are oxidized in the cells to release energy. They include sugars, starch, and cellulose. Sources of carbohydrates include cereals, root tubers, sugar canes, and stem tubers. Sugars go in cold water and have a sweet taste. Sugar is Disaccharides. Carbohydrates can be classified depending on the number of sugars as monosaccharides, disaccharides.
Okay, thank you very much.
You're welcome.
Rosemary, it is taking us to the different foods that we are going to talk about. We're talking about nutrition. So we want to see now the different foods. That foods can be carbohydrates, food can be proteins, food can be vitamins, foods can be lipids. Yeah, we can have foods as the water, we can have food, we can have refuges, okay? Like so we're going to be looking at each particular class of foods. Remember at the beginning, we are looking at what should be the component of food, and we're saying food must give you energy, food must be able to be used for building the body, food must be for protection, yeah, and the like. So we say the foods we have fall into such categories, those three, they can be able to serve those three purposes. So today we're looking at carbohydrates, which foods we are saying they provide energy. When they are broken down, hello? Hello? Hello?
Yes, teacher.
All right. When they are broken down.
Yes, teacher.
Yeah, okay, I'm back. When they are broken down in the body, we get energy. So carbohydrates, in simple terms, would include those foods that provide the body with energy when they are oxidized, okay? And the quite many sources where in our daily foods that we eat can be the cereals. You people eat a lot of cereals as a source of carbohydrate, and in a day you need so much carbohydrate. And if you eat today, if you have had posho, if you have had rice, these are most common foods at our homes. And the reason is because of their good source of energy. That's why we use them so much.
So your network is breaking.
Okay, sorry. Okay. You should maybe to improve with time, but we today had the problem of network. All right. Then sugars are like we have given root tubers. You know, cassava. You know, cassava is a root tuber. You eat so much of that. You eat so much of roots, which are good source of carbohydrates. Then sugar canes. Then many other stem tubers. You eat Irish. Irish is a very common in Uganda here. We eat a lot of Irish as a good source of carbohydrates. Now, the gift of this topic or the subtopic is for us to understand how we classify carbohydrates. Okay. And carbohydrates, we shall classify them into three. We shall have the group we call monosaccharides, and disaccharides, and then polysaccharides. Now, the classification of these carbohydrates depends on the number of sugars. So the kind of sugars we call monosaccharides, these ones, mono comes from the word one. Then saccharide means the sugar. So if I say monosaccharide, I mean those kind of carbohydrates with just one sugar molecule, okay? One particular sugar molecule. So the type of sugar is one. I can give an example of glucose. Glucose is a monosaccharide, okay? Another example I can talk about is fructose. Another example I can talk about is the Garakitos, okay? So in this case, we have covered the monosaccharides in great, and they are saying they are sugars with the general formula. That's the general formula, and for them they are the simplest sugars, as you can see. Being simple, it means when you eat that sugar, it is absorbed there, and it then doesn't need digestion. Normally, if you need the instant source of energy, you take a glucose. Reason, because it doesn't require digestion. Yeah. So when you have come from a long race or long activity and you want an instant source of energy, you eat glucose. Okay, you have seen during the sports, athletes or footballers get a good source of energy from this glucose, because as you take it in, it's absorbed directly and it gives you energy. So no digestion is needed for these simple what? Simple sugars. Okay. And one characteristic of monosaccharides, one, they are sweet, they are crystalline, soluble in water, and they're always smaller molecules. Now, in the lab, we can be able to identify a sugar which is a monosaccharide from a disaccharide and then a polysaccharide, like so. So since we're talking about monosaccharides, the monosaccharides, we can, for example, glucose, we can test for them in the laboratory. And the common test here is what we call the reducing sugar test. Now, when we get back to school, we shall be able to do experiments involving these reducing sugar tests, or these, or what we call food tests. When you do food tests, we are interested in identifying the content of some foods we are provided with in the lab. We carry out tests to analyze which food substance is it. Is it a reducing sugar? Is it non-reducing sugar? Whatsoever. Okay. Say non-reducing sugar. Is it starch? Is it a disaccharide? Because all monosaccharides are called reducing sugars. Why? Because when we test them in the lab, we test them with a reagent which is called iodine solution. And the iodine solution contains copper-2 ions. And these copper-2 ions are known to be blue. If you know the color of copper-2 sulfate, it is blue. Even copper-2 sulfate solution is a blue solution. Now, copper ions have a blue color. So even this bendic solution has a blue, it's a blue solution. Okay? Being a blue solution, it means it has copper-2 ions. So the reaction that involves testing for reducing sugars involves these copper-2 ions being converted into copper, copper 1. They are copper-2. They are converted into copper 1 oxide. Copper 1 oxide has a brown color. Okay, so that's why we are moving from copper 2 to copper 1. So we are reducing the oxidation number of copper 2 plus to copper 1. That's why we are saying reducing. If I move from 2 to 1, I'm moving from copper 2 to copper 1. These two compounds have different colors. Copper 2 ions are blue. Copper 1 ions has a characteristic brown color. So why we see a red-brown color in the reaction is because copper 2 ions are reduced to copper 1, and then we get that brown color, meaning that this reaction only occurs with any kind of sugar which has got a reducing effect. So it is the sugar which causes the copper 2 ion to be changed to copper 1. And then when this happens, we see those characteristic color changes, as you shall see them, that the solution will return from blue. Blue is now the original color of the reagent, to green, to yellow precipitate, and finally an orange or red precipitate. And when this happens, as we shall see when you carry out the experiment, it shows that the sugar is reducing what? Sugar. So monosaccharides are known to be reducing sugars because they can carry out this effect. So this reaction we have been describing is only possible with monosaccharides and some disaccharides, for example, maltose. Yeah. For example, maltose is a disaccharide, but it can act as reducing sugar. We shall see later, because it is made of 2 glucose molecules, and during its formation, it leaves room for its chemical nature, leaves room for that reaction to take place. However, other non-reducing sugars, their chemical formation, okay, their structure formation does not allow such a reaction we have been describing above to take place, and we don't see color changes when we we carry out the bare tests in that case. Okay, I maybe literally we might have got to understand what reducing sugar is. However, the interest here is for us, one, to know, should know what monosaccharides are, and then you should have examples of monosaccharides. Can someone give me 3 examples of monosaccharides? Three examples of monosaccharides or simple sugars. Hello? Hello?
Yes, teacher.
Examples of monosaccharides. One of them, Tigud has given us glucose. Okay, another one. That is correct. Can we get another? Fructose, very good. That is very good. May I get another one? Rosemary, I've talked so much today. I should choose someone else. Nachiboneka, are you there? Lydia? Unmute and talk to us.
Yes, Master.
Can you tell us something?
Good afternoon, Master.
We are in the lesson. Fine. I've been sleeping already. Anyway, very good. Eh?
It's not so boring.
Pardon?
It's not so boring like at school.
So this time maybe you are studying as you are watching the TV. The young sisters are also around. So things are not boring.
I'm eating.
Don't cause the hunger to us who are not eating. You know, when you talk about food and it's only you eating, the rest who are not eating, they start yawning, and you see the number of participants reducing, because people will be rushing people will be rushing for food. Yeah, when you talk when you think about food, that one normally happens is the like for food also increases. All right. So we have understood the examples of 3 common monosaccharides: glucose, fructose, and galactose. Always have to remember those 3 simple sugars, okay? They have to be at your fingertips, and you always understand them, because they're going to help us understand the next part of disaccharides, okay? If you miss understanding those 3, then it will be difficult for you again to understand the next part, which I call disaccharides. Now, monosaccharides are used as building blocks. They are used as building blocks for. They are used as building blocks for. They are used as building blocks for other sugars, the disaccharides and then polysaccharides. May I ask people to mute? Okay. Now, I was saying that monosaccharides are building blocks of disaccharides and the disaccharides and then polysaccharides. So meaning that we can build the other sugars we can build them from these monosaccharides. That's how I've told you to always remember those 3, because they're going to so much come across them, and we see how they are used to form other saccharides, the disaccharides and polysaccharides. Now, by disaccharide, what di means 2, and I told you saccharides means sugars. So disaccharides are a combination of 2 monosaccharides. So when you have 2 monosaccharides, you get a di what? You get a disaccharide. That one should be understood from here. Okay. For example, however, when I have sucrose, sucrose is cane sugar. Cane sugar I'm talking about is obtained when I combine glucose and fructose. These two were examples of monosaccharides. So when I combine glucose and monosaccharides, I get a sugar which is a disaccharide. And in that reaction, it's called a condensation reaction because we lose a water molecule. That's why the equation here we're saying when you get glucose plus fructose, you get sucrose. And then we produce a water molecule. The other example, so that is the cane sugar, which we call sucrose. Today, everyone has taken sugar, not so. Sugar it will come from sugar cane, sugar beet, and the like. That sugar is sucrose. That sugar is sucrose. That sugar is particularly sucrose, and is a disaccharide. The other one is maltose, which is found in germinating seeds. When you have germinating seeds, the kind of sugar there is maltose. And this one is good if you combine two glucose molecules. So glucose plus glucose, I get maltose. In a reaction which I call a condensation reaction, where water is given off. Then the other third type of disaccharide is lactose, which is what sugar which is common in milk. All of us take milk. If we enjoy milk, the sugar in milk is a lactose. Yeah. So that sugar has glucose and glucose and galactose. Okay, combined together in a condensation reaction. Condensation means you are combining two simple molecules to get a larger one. Okay, so I combine the simple molecule glucose and galactose, which are monosaccharides. Then I get lactose, okay, which is a disaccharide. Okay, meaning that my monosaccharides, glucose and galactose, are the building blocks which I have used to make another disaccharide, which is in this case milk sugar, or what we call lactose. In the reaction, water is given off. That's why it is a condensation reaction. So these disaccharides, they are called double sugars, okay? Because there are two sugar molecules we have combined via a condensation reaction. Of course, an enzyme is required for such reaction. That's why in the first case, we looked at enzyme lactase, okay? Not okay, all lactose enzymes, all lactase enzymes, okay? They are the ones required for this case. In this topic, we shall also talk about enzymes in the details, okay? So we can leave it for, you can leave enzymes for now, because we shall talk about them. Okay, may I get any questions so far about disaccharides? If anything needs to be talked about. Yes, Chigundu.
No, master.
Okay, all right. Okay. Then for that case, let us continue. If there is no rising question out of that, we can now look at the properties of disaccharides. They are sweet, like we saw monosaccharides. Milk is sweet, not so. Sugar is sweet. Yeah, these are examples. They are soluble in water. Milk is lactose is soluble in water. Cane sugar we use at home is soluble. Cane sugar is crystalline. Okay, so
Your network is breaking.
Oh, sorry. Sorry about that. Hope, is it okay now? Is it better?
Yes.
Okay. Just pray it gets better. Now, So properties of disaccharides, you have seen them as sweet, soluble in water, crystalline, and can be reducing, e.g. maltose, and then non-reducing, e.g. maltose and non-reducing sugar. Can you correct that? To maltose are non-reducing what? Sugars, not and, they are. Okay. So, however, we also have the other types which are non-reducing. That is, sucrose is a disaccharide, but it's non-reducing. On the maltose is, if I take you back to the examples of those ones we have those two there. Okay, we have those two there. Then now disaccharides can be changed into monosaccharides. If I have a disaccharide, I can change it into monosaccharide. That reaction is a hydrolysis reaction, because when they are being formed, there was loss of water. If I can add the water molecule to that compound, I mean I'm hydrolyzing it, then I can be able to go back to reverse the reaction. So it is possible to move from a disaccharide to a monosaccharide via what we call hydrolysis reaction. Sometimes we boil the disaccharide with an acid, like I can use the dilute hydrochloric acid. So if I see me boiling a disaccharide with an acid, I'm trying to cause what we call hydrolysis. I want to convert it back to its simple sugar that we are used for making it. All right, and in so doing, I'll get my monosaccharides out of it. And that is a commonly experiment that we handle in the laboratory. When we start on our practical biology, you'll be involved in such reactions. I know when you get back to school, you shall be doing such practicals for this course to understand this work better. Okay, then we can move to the next type of carbohydrate. These are the ones we call the polysaccharides. You have understood the word poly means many, saccharides means sugars. So this means the polysaccharides, they contain very many sugar units. It can be even like 1,000 mono units, monomers, or like a glucose can be 1,000 or more to make a molecule which we call starch. So right now I've given you one example of polysaccharide. The other is glycogen, and then cellulose. Starch is common in the foods we eat. Glycogen is a storage form of sugars, carbohydrate in the animal cells. Whereas starch is in plants, glycogen is in the animals. Yes, Lydia. Lydia?
Once that was a mistake.
Okay. Now, what happens is that we are looking at polysaccharides, and we have looked at the three types of We have looked at the three types of polysaccharides. I was giving you a difference between the starch and the glycogen. That whereas animals have glycogen, like in our bodies, we have glycogen. When you eat starch foods and they are digested, the glucose, excess of glucose is converted into glycogen for storage. Okay, when it is converted into glycogen for storage, in our cells find the glycogen. But for plants, where we got the starch, they had the starch in their cells. All right, meaning that starch occurs in plants, whereas glycogen occurs in the animals. Hope you have got the difference now between glycogen and the starch. Then for cellulose, it is common in the plant cell wall. A lot of the plant material we eat has a lot of cellulose. That's why I have even what we call cellulose cell wall. Cell wall of plants is made of that material called cellulose. So if you eat plant material, you eat a lot of such cellulose material, which is a source of sorry for the network, which is a source of carbo. Which is an example of polysaccharides. The other thing you know about starch is that it is an insoluble carbohydrate. Okay, then shall not dwell so much about the chemical formula in this case. Now, I want us to understand the uses of carbohydrates in the body. Yes, can we get the uses of carbohydrates in the body? Hello?
Sir?
Yes, uses of carbohydrates in the body. Yes, can I have a response from Paula?
What
Paula? Yes, Paula.
Source of energy.
Very good. It's their source of energy. Precious. Precious. Natkunda. Philo.
Stored as food reserves plants and animals.
Okay, yeah, that is very good. We have just been talking about that. How they are stored. They are
stored as food reserves in plants and animals.
Very nice. Okay, and that's what we have discussed previously about the difference between between starch and then glycogen. Yes, Shiba.
present, sir? It forms parts of that. It forms part of blood cells, wall, and other body structures.
Very nice. So it forms part of plant cell wall and then other body structures. Here I was telling you about the cell wall of plants being made of cellulose. Okay, so in that particular we have cellulose, which makes part of the cell wall. So that is the function of or the use of a carbohydrate for the plants. Someone has told us that they give support. Cellulose. Plants which have cell wall have that rigid structure because of cellulose, so it gives support. That is correct from Chigundu. I had other two hands from Alan or Emmanuel. Yes, Emmanuel. Yes, Emmanuel. Emmanuel. Okay. Okay, we can continue. They are used to manufacture other nutrients in the body, like we can get lipids from them. Okay, so when you have when you have them present in the cells, you can make other. You can make other units. Okay, you can make other units out of them. Okay. Okay. Now we have a few minutes there left to the end of our lesson, like 70 minutes or 10. Let's see how we can use the remaining part of the time to understand this. Now, testing for carbohydrates in solution or in foods, and most times we use solutions, and we can be able to test for food reserves in the solution. And for this case, if I want to test for starch, I'm going to go through. I have a thinking. We can reserve this for school, because even if we go through it and we have not been able to do it, it becomes we may not benefit out of it. Okay, I don't know what would be your say. Okay. Okay, let me talk about it. For example, if you're testing for starch, we use one iodine solution, like I've been talking about it before. And we need a test tube. We need two different solutions, of course. Solution A was group starch. Solution B was a glucose solution. Now, to 1 certain test tube of solution A in a test tube, you add 3 drops of iodine solution. The solution turns into black solution. Of course, to turn, if it was turbid, it will go into black solution. And if that happens, then it means that the starch is present. If I have the same in a different solution, I add 3 drops of iodine solution, but I get color of solution turning into brown. It means that the brown solution will be the color of iodine. The solution will have attained the color of the reagent, which means starch will be absent in this case. Okay, that is how we can test for starch. If we get to the lab at school, we shall be able to experiment these tests we shall have trials to understand these reactions. Okay.